So let me start out right with the most fascinating story. Until about 5 years ago, I thought that this bug, namely Toxoplasmosis gondii, is the most interesting microorganism that interacts with the human brain and with animal brains. Make a very long story, complex, fascinating story short. I would recommend anybody to read up this fascinating paper in Scientific American. For this organism to complete its life cycle, it has to be eaten by a cat, and the cat eats basically a rat. In order to maximize the likelihood that this rat that has the microorganism, the parasite in it, gets eaten by the cat, this organism can change the limbic system of the rat. Rather than running away and triggering a fear response, it switches it to a sexual arousal response. So this organism has managed in evolution to figure out how to, what probably most neuroscientists, very few neuroscientists even would dream about, can completely change the engagement of two totally separate systems within the brain for its own benefit. Now this was until about 5 years ago. I have to say that coming to this field, we now realize that there's a lot of organisms that are not parasites that actually live inside of us normally. 100 trillion of them, 10 times more cells than our bodies, our human cells, that these organisms are capable of a phenomenal range of things by mechanisms that we're just beginning to understand, by signaling from the gastrointestinal tract to the brain, and the brain talking back to them in multiple ways. So the way they do this is these are the microbes. This is the gut wall, the layer that separates the lumen from the outside. And by a series of metabolites, and they produce a few hundred thousands of these chemicals, different organisms can produce these metabolites to get into the blood. Here are just 4 examples, particularly well studied. One that produces a major risk factor for cardiovascular disease. And this one, 4-EPS, which has recently been shown in a study that at least in animals, can have a major effect on the brain of animals, producing an autistic-like behavior. So here's a little bit more detailed explanation of how we communicate with these microorganisms in our gut. And just keep in mind, 100 trillion, they're so small that we don't see them, but it's a lot more than us human cells in it. And they can talk to us in various ways. I've just mentioned these metabolites that they produce, get into the blood, reach the brain. They can also talk to the immune system and influence cytokine production, makes it to the brain. And I'll show you an example, show you a couple of examples. One is what influences of how many of these gut signaling molecules get to our brain, into our circulation. And then also, particularly interesting cell that produces serotonin in the gut, that these microbes have a particular affinity to. So this is a general concept. A lot of people have heard about the leaky gut. This has been amazingly a lay fascination long before scientists and gastroenterologists came up with that this is actually something scientifically valid. The leakiness is basically the space between 2 cells that make the inner wall of the gut. And there's various factors that can influence this leakiness between these cells, which then allows these metabolites to get in much larger numbers into our bloodstream, and then including reaching the brain. So I see the wrap up. This is actually pretty scary. So I have to, let me say a couple of words about this particular cell, the serotonin cell. And for psychiatrists, obviously this is shocking news that 95% of the body's serotonin is in our gut, not in the brain, even though psychiatric drugs are thought to act through the brain. And not only that, but they're regulated, so we know now they're regulated by these gut microbes talking to these particular cells. And this is an illustration of that. You see one of these cells that contains serotonin in the gut wall. This cell forms a synapse, we just learned this in the last year, with sensory nerves like the vagus nerve, and basically now can talk back to the brain, and microbes that interact with the one end of this cell can stimulate both the production and the release of serotonin, which then acts back into these limbic brain centers within the brain. The brain in turn can also has a way, these cells are also innervated by the sympathetic nervous system, so the brain can regulate the release of these. So if you think about this, this is really an extension of the brain's serotonin system or the emotional system. And now we have another partner in this conversation, not just the environment or the person next to you, but it's the microbes sitting there that constantly talk to, 1 minute. I have to go through these slides because, I mean, it's kind of a fascinating story. So most of the things that we have learned about that microbes can actually influence emotional behavior has come from this experimental setup. Germ-free mice, so mice that have grown up in a completely sterile environment. And you see this on one side, and people have compared the germ-free mice with normal mice and have characterized their behavior. Now these mice that are germ-free, that have no microbes inside of them, are more stress reactive and are socially withdrawn, autistic-like behavior, whereas the other ones have more pain and are more anxious, the ones that have the microbes. Another example from these kind of studies, equally amazing, that you can actually transplant the behavior from the normal mouse, just taking the fecal pellets, the stool of that mouse, into a mouse that doesn't have its own microbes, but has a genetic background that actually makes it extrovert and gregarious. So what happens with that germ-free mouse that is normally the outgoing one becomes timid and introvert and anxious by itself, simply by transferring the stool and the microbes that are in the stool from one animal to the other. Third example, particularly relevant to our time today. So the normal mouse is actually not quite normal. It's a so-called knockout mouse that lacks a particular receptor in the brain that interacts with microbes in the gut. These mice are obese and they like to overeat. So if you take the stool sample from these mice into, again, this mouse without any of its own organisms, that mouse not only becomes obese, but it starts to become hyperphagic or overeating. So simply by transferring whatever is in the stool samples in the microbes and their metabolites can transform or transduce this trait. Based on these remarkable observations in the last 5 years or so, or 7 years or so, people have come up with speculations about a range of psychiatric diseases. And look at my psychiatric colleagues who are obviously shocked by the fact that the GI tract may be relevant in psychiatric disease. But this has been postulated. Unfortunately, the human studies are just beginning. There's a large number of large-scale studies going on. I think one that has received, in terms of obesity and food addiction, that's probably best established. But autism is one that has received a lot of attention. It's probably not as simple as just doing a fecal transplant and curing a child with autism spectrum disorder. But there's convincing animal experiments that suggest it may play an important role. Can I finish the last 3 slides? So I have to show this because our group was the first one that was able to demonstrate in humans for the first time that you can actually, you have this communication in humans as well, not just in these nice-looking mice. And so what we did a study where we gave healthy women yogurt, probiotic-enriched yogurt, and we were interested, does that change the brain to emotional stimuli? And so we gave these individuals a task where they had to look at emotionally balanced faces, you can see, and match which ones are the same. I won't get into the details of this. And at the same time, we looked at their brain activity with fMRI. And you see a whole range of brain regions that were differentially activated between people that had the yogurt. It was a randomized study, and they did not eat it for 4 weeks. And you can see that this network, the way it was connected, behaved totally different in those 2 conditions. So the first example, we don't know if this at some point will have an effect on actual emotion, but the emotional reactivity that we've demonstrated in this is so far the only study that these mouse experiments can translate into humans. So I think I'm going to stop it here.